Hello, and welcome to a quick tour of the possibilities for your final assessment simulations. This is the fruit fly mating simulation. We want to enter as a guest, and then notice they prompt you and tell you what to click on as you go through for the most part. You have lots of options. You can do any of these kinds of traits. So let's try eye color. We want to choose a female with a particular eye color. So let's go female brown, add to cart, and now we got a male who's wild type. Awesome. Add to cart. And this is our shopping cart. We have a female with brown eyes and a male with wild type eyes. Okay, check out. Yes, I want to order these flies. Unpack. Into the mating jar they go. Okay, an incubator. They gotta do their business. Takes a little while, like a couple weeks. Cool. Guess you can check out more detailed information if you want. Click on the jar. Open the incubator. Okay, here's cross number 1. And it tells us again what our female and what our male are. Click on it and knock them out. This is what, scientists would actually do because it's kind of hard to count up fruit flies that are moving around. We're under the microscope now. Start our flies. And it tells us what the results are. If you hover over, it tells us we've got the female and this is the type. Here's the male and this is the type. So it tells us the numbers of offspring and it tells us what they are as well. Awesome. Now we can actually use these to do a second cross. So I clicked on the pile, it brought me up close and personal. This is a female wild type, and I can use a new mating. Fly added to new mating jar. Awesome. Now I have to zoom out and I have to get my male. So the other pile, cool. Use a new mating. Okay, now I have a female and a male in the new mating. Now I want to go back to the other screen. So zoom out, return to lab. Voila, look, this is my new cross. So I have to mate them again. And wait till they're ready. Awesome, they're ready. Open it up. Notice we've got mating jar cross 1, mating jar cross 2. The most I ask you to do for the final assessment is 2 crosses or 2 generations. So this is first generation, this is second generation. Let's click on it. Sort them up again. Awesome. Now this is my data for my second generation. So that is one set of 2 crosses, 2 generations, boom, done. You have to do a total of 3 of those for this simulation. Hope you enjoyed this one. Let's move to the next. And now, welcome to mutation simulation. This should look very familiar because I had you play around with this when you were learning about genetic modification. So again, same protein as we used before. This is the starting protein. This is the baseline. But what you can do is you can edit that DNA. You can add bases in. I asked you to do a multi-point insertion. I could add 1 base. Well, no, that wouldn't be multi-point. 2, 3, 4, 5, 6, 7, 8, 9, 10, as many bases as I want. Those are all multi-point insertions. I also ask you to delete a base in a few places and see what happens. Just pop in, take 1 base out in a couple places, see how your protein is affected. And then the third one that we ask you to do is a substitution. So go in and substitute 1 base, so a single point substitution in multiple places, and see what happens. Then when you make those changes, you hit apply. We didn't make any changes, nothing's going to change, but you hit apply and you see what happens to your protein. Reminder, in between changes, what I would suggest that you do is you refresh. Go right up here, hit this refresh button, because if you don't, sometimes it seems to get a little messed up. But what we ask you to do is collect 9 different data points. So 3 single point removals, 3 single point substitutions, and 3 multi-point insertions for your final assessment. Think about it. Maybe this is the one you want to do. Let's check out the third and final. And here we are at the protein folding simulation. I have to admit, I have quite a fondness for this one. The first thing you have to do is hit play at the bottom. Then you can see the starting protein. Notice it's nice and big, so it's fun to play around with and to do stuff too. Well, let's see. Right now we're looking at hydrophobicity. So this tells us which ones are hydrophobic. These are the tan or brown ones here. And it tells us which ones are hydrophilic, the green ones. You can also tell if you click on it. It gives you a reminder. Each each amino acid, because each of these circles represents an amino acid that you click on, will tell you its name and will tell you that it is hydrophobic or hydrophilic. Brown, hydrophobic, green, hydrophilic. Okay, so the cool thing here is that you can take it and change the environment it's in. So this is a mix of hydrophilic, hydrophobic, right? This is water-based. You can change this to oil. You can also change this to a vacuum. Great. Let's go back to water, starting one. Then what you can do is you can change the protein. So we can change it to all hydrophobic. Okay, so now we're all water-fearing. We are in water. You can change it to oil. You can change it to a vacuum. So you can play around with what happens here. Finally, you're going to want to go all, not finally, but another thing you can do is go all hydrophilic. And again, move it from water, oil, vacuum. Awesome. Then you can do things, let's go back to water here. Then you can do things like make it mostly hydrophobic and mostly hydrophilic. So the basic idea here is that what you are doing is changing these, what the amino acids are, what the properties of the amino acids are, whether or not they're hydrophobic or hydrophilic, and you're changing the environment that it's in to see how that affects the protein. So we ask you to gather maybe 12 data points, I think it is, pictures of the proteins, and then to make a claim about how being hydrophobic or hydrophilic affects it and how this environment piece affects it. Good luck, and I hope you've enjoyed this tour of these 3 simulations and hope you have an idea of what you want to do. Please come see me if you have any questions.